Hello everyone, my name is Nabila Anindya Salma. My student number is 6200022823. I am from Biology Department of Chulalongkorn University. I would like to present you about Biocontrol Antifungal agent potential of Bacillus subtilis SCB-1 for fungal phytopathogens. My advisor is Dr. Krieng Kanchanawatee. The outline for this presentation divided into 5 sections. The first one is the introduction, the second one is methods, the third one is result, the fourth one is discussion, and the last one is conclusion. First, what is fungal phytopathogens? It is a fungal which can cause negative effect to their host. That fungi process a wide range of enzymes, destroying the carbohydrate polymers which constitute the building materials of the cell walls. There are several examples of fungal phytopathogens which is very common in the environment. If you can see, figure number 1 is Scrofularia lunata, figure number 2 is Phomopsis sp, and the last one is Fusarium oxysporum. Fungal pathogenic microorganisms become major threat to the stability of the agriculture and ecosystem. Why? Because they threaten the production of crops that are already growing in the field and can cause post-harvest disease. As you can see in the figure number 4, there is an infection of fungal in several leaves. And then in figure number 5, 6, and 7 is a fungal disease in sugarcane. The fungal pathogens make a black spot or brown spot which can killing the whole leaves. To combat the fungal infection, farmers rely on resistant crop varieties or multiply fungicide treatments, which can have negative effect on the environment, like first, affect soil organism and the crucial function of this organism are responsible for, and then it have a high toxicity, and then it have lasting impact on the fertility and health of agricultural soils, and last one, it have the potential to cause adverse effect to the structure and functioning of aquatic ecosystem. That's why there are alternate measures to combat fungal pathogens. It is from the endophytic bacteria, and we can use it as a biocontrol because it has several effects. Like first, it stimulates the growth of plants, and then it has antimicrobial compounds production, and then it shows antagonistic potential against fungal phytopathogens, and then they have been reported for their beneficial effect in crop health, and then it can use as a plant protection against pathogens and disease, and last but not least, it is improve of ecological adaptation of the host. I will separate my method into 3 sections. The first one is isolation and identification, the second one is bacteria screening and identification, and the last one is what produce isolate SCB-1 to inhibit the fungi. First section is isolation and identification. First method is isolation of the fungal pathogens. So they collect infected sugarcane and then they incubate into the Potatodextrus agar. After that, they incubate it and then they got the heavy tips of the fungal colonies. They subculture into Potatodextrus agar to get a pure culture. After that, they move into the identification of fungal pathogens. So the fungal pathogens isolate, identified using PCR. After that, the major pathogens were sorted and used for the further studies. Here is the result of the identification of fungal isolates. Only 10 fungal isolates from sugarcane pathogens that they use for further studies. The second one is bacteria screening and identification. Then the isolation of the bacterial endophytes. First, they collect uninfected sugarcane from its leaves and stem. After that, they isolate it into tryptic soy agar. After that, they move into screening for antifungal activity method. So they do the dual culture technique in Mueller-Hinton agar. They put bacterial in this side and the fungal isolate in the opposite side. After that, they incubate it. After they incubate it, they calculate the zone of inhibition using the formula. The percent of inhibition equal A minus B divided by A times by 100. After that, among 7 isolates, only 1 isolate SCB-1 that showed antifungal activity against fungal pathogens. And then here is the result of the antagonistic activity. This side is a fungal isolate and the opposite side is a bacterial isolate, as we can see in the red mark. As we can see in the control culture, the fungal isolate can grow to all over the plate, but in the dual culture, there is a zone of inhibition between fungal isolates and isolate SCB-1. It means that there is an antagonistic activity between bacterial isolate SCB-1 and fungal isolates. And then this is the result for the distance of inhibition against 10 fungal isolates. Fungal isolate SC5.1, or called Curvularia lunata, has the highest inhibition distance, and then fungal isolate SC8.1, or called Fusarium verticillides, has the lowest inhibition distance. The next one is the characterization of endophytic bacterial isolate SCB-1. The bacterial isolate SCB-1 identified based on the sequence of 16S ribosomal RNA genes. After that, the sequence was finally submitted to GenBank and occasional were obtained. After that, they make the phylogenetic trees. Here is the result of the identification of the bacterial strain with antifungal activity. This is the picture of phylogenetic tree. The sequence of isolate SCB-1 is evolutionarily related with Bacillus subtilis strain 80cc. From the morphology, confirmed that bacterial isolate SCB-1 is Bacillus subtilis. The last section of the method is what produce isolate SCB-1 to inhibit the fungi. They do the preparation of culture supernatant and metabolic extract. First, bacterial isolate SCB-1 growth overnight into Luria bertani broth. After that, they centrifugate it. After that, they cut the supernatant, and then the supernatant filtered with Whatman filter paper. After that, they extract with the volume chloroform and methanol one by one. After that, the chloroform soluble spread and concentrate under vacuum. After that, it dissolved in methanol or dimethyl sulfoxide, or called DMSO. After that, we move into antifungal assay method. 5 mm of mycelial plugs from the growing fungal culture were put into the center of PDA plates. Then, agar that already plugged into bacterial extract put in the side, and agar that soaked in control the MSO put into the opposite side. After that, they incubate it. After that, the percentage of growth inhibition calculate using the percent of inhibition equal A minus B divided by A times by 100. A is denotes the growth diameter of the fungal in control medium, and B denotes the growth diameter in culture medium. And then here is the result of the antifungal activity of the culture supernatant again for test fungi. All of the control wells were covered by fungal hyphae, and all of the hyphal growth was inhibited towards the wells that load with bacterial supernatant. Here is the graph that representing the colony diameter of fungal isolate with different volume of chloroform and methanol extract of bacterial isolate SCB-1. The chloroform and methanol extract organic fraction showed inhibition against all the selected fungal strains. As we can see in this graph, if we put more volume of chloroform and methanol extract until 500 ppm extract, there is a strong inhibition against all the selected fungal strains. Now they know that bacterial isolate SCB-1 produce something. So next, I am going to talk about what bacteria produce to inhibit the fungi. They test the bacteria strain using starch agar for tested alpha amylase activity. They test the bacterial strain using carboxyl methylcellulose agar to test cellulose activity. They test the bacterial strain using Kingsby agar for test chitinase activity, and then they test the bacterial strain using skim milk agar for test protease activity. Presence of clear zone considered as positive, and then the result is there is a presence of clear zone among 3 tested plates only. There is only positive for alpha amylase activity, cellulose activity, and protease activity, but there is no chitinase activity. And then they do the assay of antifungal volatile compounds. They put bacterial isolate SCB-1 into the nutrient agar, and they put 5 mm mycelial plugs into the potato-rich rose agar. They did the same thing for a control, but without bacterial isolate SCB-1 in nutrient agar. After that, the PDA plates inferred over the NA plates, and then they measured in 24 hours in 5 days. Here is the graph of the percent inhibition of fungal isolate from the volatile component inhibited assay. After 48 hours, there is significant of percent inhibition. It means that the antifungal volatile compounds were produced by the isolate SCB-1 could inhibit the growth of fungal colonies. Then they do the detection of secondary metabolic biosynthetic genes. The Bacillus subtilis has been reported to produce different secondary metabolic biosynthetic genes. 8 selected genes were selected for screening through PCR-based method, and then the amplified product was analyzed on a 1.2% of agarose gel. Here is the result of the antifungal biosynthetic genes in agarose gel. Among 8 kinds of genes that are tested, only 5 genes that appear in the agarose gel. There are BEER genes for bacillin biosynthesis. There are D, F, and D gene for diphyscidin biosynthesis. There are SRFA genes for surfactin biosynthesis. There are MLNA gene for macrolactins biosynthesis, and there are FENA gene for fungicide biosynthesis. Bacillin has antagonistic activity against fungi, bacteria, and antibiotic. And then for diphyscidin and macrolactins, it is a broad spectrum antibiotics. They are a group of polyketides. Polyketides is a large group of secondary metabolites that contain carbonyl and methylene. And then for surfactin, it has antifungal, strong hemolytic, and surfactant properties. And then it has antimycoplasma and antiviral activity. And then for fungicide, it's potent for antifungal agent. They are group of lipopeptides. Lipopeptides is a acyl peptides that contain an alphatic chain with amphiphilic structure that produce as secondary metabolites for microorganisms. Then they identify the bioactive component. They extract from the bacterial-free supernatant, separate using UPLC. After that, they detect the fraction, and then the ASE KTOF mass spectrophotometer connect to the UPLC system. Here is the result of the analysis of the antifungal compounds. The LC-ESE-MS analysis of the bacterial extract indicate the presence of an antifungal compound called surfactin in the retention time of 12.45 minutes. And then here is the result of the mass spectral data that revealed the presence of surfactin C15 with an M or Z of 1037.3. After that, they do the determination of biocontrol potential of bacterial isolate SCB-1. They have 2 treatments. First, the treat seed that already soaked overnight in bacterial isolate SCB-1 suspension. After that, they soak again into the spore suspension for 30 seconds. After that, for the untreated seed, they soaked overnight in 0.85% of sodium chloride solution. After that, they soak again into the spore suspension for 30 seconds. The spore suspension consists of Fusarium oxysporum, Fusarium verticilloides, and Fusarium sp. The result between untreated and treated germinating seed after 48 hours of fungal inoculation are the SCB-1 treated and untreated seed control are successfully to germinate, but all of the bacterial isolate SCB-1 untreated that already soaked in fungal suspension failed to germinate, but all of the seed with SCB-1 treated which already soaked in fungal suspension successfully to germinate. Here is the result from the graph that representing the germination percentage of mung bean seeds. We can see that the two, control and SCB-1, doesn't have any effect of germination. If it's lower, we cannot use it. For untreated seed with SG7.1, SG8.1, and SG9.1% germination, very low, it's about 0. But the seed SG7.1, SG8.1, SG9.1 with isolate SCB-1, we can see that could rescue the percent germination about 50 until 70%. And then for the graph that representing the root and shoot length of germinating seed, we can see that in the graph, we compare the control and isolate SCB-1 bean growth better with isolate SCB-1 and increase the growth of root and shoot length. And for the untreated seed, SC7.1, SC8.1, and SC9.1, the growth rate reduced to 0. It means they die. And again, treated seed SC7.1, SC8.1, and SC9.1 with bacterial isolate SCB-1 could rescue the length, not like normal, but at least 50% of than the control. After that, we move into the discussion part. The supernatant isolate SCB-1 showed negative chitinase activity in the chitin agar medium, but the supernatant isolate SCB-1 showed positive activity of alpha amylase, cellulase, and protease activity. Although the chitinase test showed negative result, the culture supernatant showed antifungal activity. This concludes that the antifungal activity of the bacterial isolate SCB-1 is not because of chitin. And then, in the previous studies, many bacteria digest chitin to degrade the cell wall of fungal pathogens. But in this study, Bacillus subtilis, or known as bacterial isolate SCB-1, cannot produce chitin. The antifungal activity is not because chitin, but it's because another volatile components and lipopeptides. After that, among the 8 genes previously assumed to be present in the Bacillus subtilis of SCB-1, only 5 genes that showed in the electrophoresis. This concludes that the Bacillus subtilis SCB-1 strain cannot produce bacillicin, iturin, and bacillibactin. In this study, the antifungal activity from mass spectra activity caused by surfactin C15 activity only. Among the 3 well-known isoforms C13, C14, and C15 surfactin, the C15 surfactin has been reported to show highest surface activity. The surfactin activity could disrupt the membrane stability of fungal pathogens. After that, we move into the last part, the conclusion. Bacillus subtilis or SCB-1 has antifungal activity. Antifungal compound that produced by Bacillus subtilis SCB-1 such as surfactin and other volatile compound which is promising to be applied as biocontrol for fungal infection. After that, here is the reference that I used. Thank you for your attention. If you have any question, please write down below in the comment. Bye.